may need to wait for a couple of seconds. Right, good morning and welcome to the 8th in our series of Marine Tech webinars. My name is Mandy Bowden and I work for the Marine Business Technology Centre, which is a Plymouth City Council led European funded project. I'll be your host for this event. During this webinar, you will only hear myself and our presenters speak, and we will not be able to see or hear any of the delegates. After the presentation, we will be holding a question and answer session. Please do use the question and answer chat bar to ask any questions as the presentations go forward. And if you see any questions that other people have raised that you'd be interested in hearing the answers to, then do give it a like, and that will send it to the top of the pile and get the most attention at the end of the presentations. Now, this webinar is being recorded with the intention to be available to others on demand. Enjoy the webinar, and I want to hand you over to Professor David Sims, Senior Research Fellow at the Marine Biological Association, together with colleagues Dr. Michael Cunliffe and Dr. Rowena Stern. So, David, if you'd like to share your screen, I'll get that one up for you.
Okay. Can you see that okay?
If you just wait for 2 seconds, I'll get you there as well. Send live. Right, thank you very much.
Okay, brilliant. Yeah, thanks for the opportunity to talk at this Oceansgate Marine Technology webinar series. Maya, our head of communications at the Marine Biological Association, was supposed to head this presentation, but lucky for her as well, she's out filming actually in Plymouth Sound at the moment for a virtual field course, courtesy of the COVID-19 situation on our research vessel. So anyway, look, we're going to present to you 3 case studies of advanced technologies that are happening in the field of marine biology at our lab. Before we get into those, I'll just give you a brief overview of the Marine Biological Association. We're situated at the prime position on Plymouth Hoe, overlooking Plymouth Sound, the sort of 1,000 square kilometres of unrestricted protected waters. But our research isn't just focused here in this part of the southwest. It stretches out into international waters. It really has a true global context. The Marine Biological Association has a long history. It was founded in 1884, and in 2013 we were awarded, in recognition of our long and eminent history and status within the field of marine biology, a Royal Charter. And this really sort of is evidence of our history and our sort of preeminence in the field of marine biology. Evidence perhaps from 12 Nobel Prize winners having worked at the lab. And it's also the accolade of being the longest established marine laboratory in the UK. So our fisheries independent surveys actually were some of the first research that was set up from the MBA when it first started in the late 1800s. And these fishery independent surveys were some of the first to be carried out in Europe. And in fact, we've carried on these surveys over the last sort of 100 years or so. They were very much our foundation, and they still continue to this day. In fact, they provided some of the, back in the 1950s, some of the first evidence of biological responses to climate change. Our vessel, MBA Sepia, you can see it there, also collects plankton, as well as environmental DNA, these small sort of strands of DNA that are shed into the water from organisms. And that allows us to study everything from viruses to giant sharks in a single water sample. We've also got world-class lab facilities. Here's an example of the advanced microscopy that we have at the MBA. It's the only mesolens microscope in any biological laboratory worldwide. And it's remarkable in that it allows wide field, low magnification images to be obtained with the resolution of a high magnification microscope. And we're applying this to study sort of subcellular detail and physiological processes. So it really is an amazing way of looking at a whole population of cells, diatoms, for example, at real high magnification to resolve the details that are happening within cells. The major new microscopy centre, the so-called Huxley Centre, allows our researchers some of the most advanced microbial imaging capabilities in the world. So it's a real sort of boost to Plymouth to have these amazing facilities right there. Also, we've got very much the latest technologies at the lab, but also still we have this sort of historical sort of talent in taxonomy, world-class sort of taxonomy from our plankton analysts. Some things are still done best by eye, and all of these different organisms you can see here can be identified in an instant by the continuous plankton recorder plankton analysts. And they've got real identification skills. And one of the case studies that we'll talk to you about is the continuous plankton recorder, which we'll come to in a few moments. We also have, as well as thinking about plankton, we also have global collaborations on other organisms, these large megafauna, marine megafauna, sharks. And we'll also tell you about research involving over 150 scientists in 26 countries, relating the fishing effort to the distributions of these exploited species. We also curate an extensive data at DASH, the Archive for Marine Species and Habitat Data. And in fact, some of their records go back to 1743. This data is accessible to all and supports delivery of policy on everything from marine conservation zones to the UN Second World Ocean Assessment. So very much sort of not just centred on the UK, but also has international scope and vision. We can't forget about the books, can we? You know, in this digital age, whatever happened to books? Well, at the Marine Biological Association, we have the National Marine Biological Library, which is one of the largest marine reference libraries in the world. Thousands of volumes of not just books, but journals. And it also holds expedition reports and even a herbarium, these sort of squashed seaweeds and other algae. MBA members can make full use of the collections and incredible facilities, including electronic journal access in-house, the new Southwood reading room you can see on the left there, a very relaxed space. I mean, you have to be careful not to sort of fall asleep there whilst you're reading your tome, because it's so relaxing actually. It's a quiet space, and actually also, as you can see there, has amazing views of Plymouth Sound. So it's a really good space to be in to conduct your learning and knowledge. We're also a learned society, and that supports the lifelong journey of marine biologists. It's through the learned society that the MBA and through its members has its voice internationally, and it represents marine biologists from 38 countries and 6 continents actually. And the quarterly magazine, The Marine Biologist, is one of the many benefits of being a member. So if you'd like to become a member, or if your company would like to advertise in the magazine, for example, please do contact us, and we're glad to hear from you. So to get on to these 3 case studies that I mentioned, as Mandy introduced, there are 3 speakers. Originally, David Johns was going to talk to you about the CPR survey. He's the manager of the CPR survey, but in fact Rowena Stern has stepped in to talk about that, and then Michael Cunliffe will give an overview of cell and molecular marine biology at the MBA, and then finally I will just give you a roundup of some of the work we've been doing on biologging of ocean predators. So if I can hand over to Rowena to talk. I'll carry on with the slides, but if you can just talk to the slides, Rowena, that would be great.
Thank you, David, and hello everyone. Thanks for listening to this webinar series. I'm going to talk to you about the continuous plankton recorder survey, which has just now clocked 7 million miles of plankton surveys, and how we're keeping this historical time series relevant. So next slide, please. Sorry, David, next slide. Okay. Thank you. First of all, why are we looking at plankton? It's thought that the healthy, functioning phytoplankton community is a nature-based solution for climate change mitigation. So we really need to understand their potential in this role to plan for climate change. Now, phytoplankton produce 50% of the world's oxygen, so every second breath that you breathe is produced by phytoplankton. And they play a key role in a process called carbon sequestration. So what happens here is that phytoplankton draw down a large amount of atmospheric carbon dioxide as they're carrying out their physiological processes. Which in turn feeds zooplankton, which eat phytoplankton, and it's captured in the zooplankton bodies. And there are a large amount of zooplankton. They are one of the most abundant organisms on the planet. And a lot of the zooplankton, when they end their life, ends up sinking out of the upper layers into the ocean sediment, and therefore drawing down the carbon away from it being reprocessed back into the atmosphere. So they really are very important for climate regulation. But they're also useful as indicators. They have a short lifespan and sensitive to environmental change. They are at a key position in the marine food web. They are at the bottom of the food web, and all other higher life forms need to directly or indirectly depend on them for food. And many commercial species start life in the plankton, such as crabs or cod. And the plankton also contains many harmful species as well, such as pathogenic bacteria, and can have a detrimental impact on marine life and humans. But generally, it's not really an exploited resource at the moment, but it's essential for the Earth's functioning. Next slide, please. How can you look at plankton over a large scale? Originally, the Continuous Plankton Recorder Survey was devised to solve a problem with herring catches and to investigate watercolour changes that were happening around the British Isles around the 1930s. A very clever chap called Sir Alistair Hardy invented the CPR device, and it was first towed from the commercial vessel, the SS Albatross, in 1931 between Hull and Bremen. Now it can be thrown off vessels doing 20 knots at sea, which is incredibly fast. And so the CPR device, you can see it's metal, it has to be very robust to withstand those speeds. The CPR survey in the UK is a global hub. However, we have sister surveys all over the world, including Australia, New Zealand, Cyprus, Japan and Brazil. Working together, we share ideas and meet regularly, and including keeping up to date with current methods. Next slide, please. So where have we collected samples? As you can see, all the red lines indicate the North Atlantic CPR survey, and you can see it pretty much covers the whole of the North Atlantic, all the way from southern Spain all the way up to the Arctic regions. But we have sister surveys. There's one going from Vancouver in Canada all the way to Hokkaido in Japan. We have a number of surveys circumnavigating Australia. You can see the purple lines, and the Southern Ocean in the green lines. So we're really truly global. We've towed 7 million miles, and we've collected over 270,000 samples and analysed them. Next slide. So what do we look at? If you look at the pictures, there's a variety of organisms that we look at, anything from tiny algae called coccolithophours, which are 10 microns, to relatively large organisms such as this squid that we found in the Labrador Sea, which was not even plankton. And the largest I think we've ever seen was a 30-centimeter pipefish, which accidentally got sucked into the CPR recorder, unfortunately for it. We in fact analyze over 700 taxonomic entities, many to species level, which is the smallest unit of identification, and a great many varieties. And there's a large team that supports this effort. So we have 10 plankton analysts, 4 members of our operation teams, 6 researchers and 1 data manager. All samples, including those that are unanalyzed, are kept in a storage site off-site, carefully looked after there in certain cold temperature conditions and in the dark to preserve those samples. Because the CPR survey uses the same methodology since 1930s, we have kept a consistent methodology, and that is a key to maintaining a strong time series, because the method is virtually unchanged since those times. The changes that we see are real and they're not due to changes in methodology. But we can add new taxa depending on the need, either as new to the survey or to find a taxonomic resolution, depending on our research questions. Next slide. So what else can we do? If you look at this picture, we have a number. The CPR survey has space in the device, at the back of the device, for a number of additional sensors and sampling devices. So on the right hand side you can see something called WAMS, which is a water and molecular sampler. This is a device that can fit in the back of the CPR that can pump water in, and I'll talk to you a little bit about what we found with using this device. But we've also deployed a number of sensors, such as a CTD, which measures temperature, pressure, conductivity and fluorescence. And we've used these plank tags, which are normally put on seals, to measure similar parameters. The CPR has a little small hole which seawater can enter to, and it is collected on a CPR internal device. It's a sort of a silk, a device that contains silk, and all the plankton collects onto that silk in a which is rolled along, so we can tell when the sample was collected, and that's how it works. So if you go on to the next slide. Now I was talking to you about the water sampler. What we do is using those. We'll talk to the water sampler in a little bit, but the CPR samples, once they've been collected and analysed, what we can also do is do additional genetic studies on samples, because we've got a sample archive dating back to 1958, and all those red dots are where we've actually extracted DNA from those samples. Sorry, if you can move back a bit to the former slide. And yeah, so what we can do with those samples is extract DNA, and researchers have now looked at long-term. If you can move back to the former slide, please. Researchers have identified bacterial pathogens. So this one is called Vibrio, which can cause cholera in some cases. And what they've done is they've looked at 50 years of cholera, and they found that the bacteria that causes cholera has increased in waters because it likes to live in warmer waters, and the number of bacteria are related to the number of human cholera incidents. So there's clearly a link there that we need to investigate further. Next slide, please. Yes, so moving back to that water sampler, we deployed this over a 5-year period in the English Channel. We carried out a 90-mile transit from Roscoff to Plymouth, and we collected water at 5 sampling points between those two sort of towns, and we tested we looked for 4 different harmful plankton types. If you look at those maps here, each map depicts a separate harmful plankton entity. The red, the circles and the size of the circles represent the amount of that plankton entity that we found. All of these plankton entities are called harmful algae. They can be harmful to humans and or animals. As you can see, each different colour represents a different harmful algae, and they all have a different pattern. And some of them are sort of more coastal, some of them are more abundant. And really, what we can really show here is that this can be applied to any type of target that you want to look at, be it sort of a nutrient or any kind of molecular or taxonomic entity that you want to look at. And by sampling over this period of time, you can get an extensive spatial idea and temporal scale of what you want to look at, and this can be potentially very useful and a cheap and effective way of measuring your target in local English waters. So if you're interested in this kind of thing, please contact me. Can you move to the next slide? Now, we mentioned about a CTD, so they measure temperature. This is an example of a temperature logger that was deployed on a CPR sample in the Pacific Ocean, and this was a very timely event because the temperature logger managed to measure what was known as quite a large phenomenon known as a warm water anomaly in the Pacific Ocean that occurred around 2013 and sort of ended around 2016-17. It's not normally only this kind of event. It was picked up by satellites, but using our continuous plankton recorder device, it was also picked up in situ, and the water temperature was able to be taken all the way from offshore in Pacific waters. And what we found was, if you look at where the arrows are, there was an increase in temperature. If you look at the green line and the red line, if you compare July 2011 to July 2013, the temperatures were quite similar. But in August 2011 and August 2013, you can see that in 2013, the temperature had really increased. And by September 2013, the offshore water was 4 degrees warmer than in September 2012. This kind of in situ measurement is really important because sometimes satellites cannot pick up these water temperatures due to cloud cover. Next slide. So last slide, we're really going to talk about how we apply our data sets. The CPR data really comes into effect in informing policy and providing societal needs. So the CPR data is often given to CFAS, a marine online assessment tool, up on the top, which is a way that we can measure our progress towards good environmental status. And plankton is used as one of many indicators in this assessment to assess the health of our seas. And this tool reports to other policy and intergovernmental organisations. So one of them is OSPAR, which is the Intergovernmental Cooperative Organisation that protects the marine environment in the Northeast Atlantic, which really came out of the Oslo and Paris Conventions to really make sure that we're keeping our seas healthy from human activities.
Sorry to interrupt, but we've got 2 more case studies. Are you getting close to the company?
Yeah. Okay. So we're just finishing now. So just to say that our data is really important for translating into policy and societal needs. That's me finished. Thank you.
Super. I shall send Michael live.
Hello, so my name is Michael Cunliffe. So as David mentioned, I work here at the MBA, and I've been asked to give you an overview really of what we do in the cell and molecular department at the MBA. So can I have my first slide, please, David? Or my second slide, sorry, if you move on. Yeah, next slide, please, David. Okay, so no, we're jumping around. So the second slide in my seat. Yeah, that's it. Thanks, David. Yeah, just stay there. Thank you. So the cell and molecular department at the MBA is made up of 5 research groups. It's Venki Modipalli, there's my group, Catherine Helliwell, Colin Brownlee and Glenn Wheeler, and together we make up the cell and molecular group. 3 of us have got joint appointment with universities. I'm with the University of Plymouth and Catherine with Exeter and Colin with the University of Southampton. And these are sort of strategic appointments to make collaborative links. And our group is funded from a range of sources. So we have 2 ERC grants, UKRI and various other agencies as well. So next slide, please, David. Okay, I mean, we're very tech focused and that's important, but David mentioned this earlier, but I think I want to reiterate it really and say how important it is for what we do in cell and molecular and how important it is to be at the MBA in Plymouth, and that is our direct access to the sea. And so I don't know how many people in the audience are Plymouth people, but if you're not from Plymouth, we really sort of take for granted being able to access the sea and being able to collect samples effectively. So David's already mentioned MBA Sepia, that's our research vessel. It goes out locally and we can get samples back into the lab within hours or within the hour, and that's really important. It really means that we can very effectively collect samples and bring them into the laboratory. So can I have the next slide, please, David? Next slide, please, David. Thank you. So, I mean, we collect samples locally and we bring them into the lab. But I mean, also, as David mentioned, and we work globally as well. And so one of the big things that we do in Cell and Molecular Group is we actually go out in the marine environment across a range of locations. We collect environmental samples and we use them. DNA, mainly DNA, but also RNA-based approaches to study biodiversity. And that's studying microbial diversity in the marine environment, but also environmental DNA, as David also mentioned. Can I have the next slide, please, David? Okay, and this is just really to give you a sense of the reach that we sort of have in the cell and molecular group. So each of these little dots on the on this world map are sample locations where we've had environmental samples collected from, and this is cruise tracks of cruises that people from someone in cell and molecular group has been on, or samples that are provided by collaborators. So we're working locally, but also globally. So next slide, please, David. Okay, so one of the really core activities that we have in Cell and Molecular Group is really using a range of living resources to do our biological research. So the MBA has maintained now for several decades a phytoplankton culture collection with hundreds of phytoplankton cultures that are used for research purposes. The MBA has recently established a marine fungi culture collection for studying both applied and fundamental aspects of fungal biology. Very recently, the MBA established a collection of essentially early diverging animals like anemones, this nematostella in this image, and we now have the culture facilities to keep them. Not technically within the Cell and Molecular Group per se, but the MBA also has expertise in kelp cultivation. This is Dan Smales' group, and also Alex Harvey, who runs the Seawater Hall, has a lot of expertise in maintaining cephalopods for biological experiments. And the history of cephalopod biology at the MBA goes some way back. And for those of you that have visited the MBA, you might notice a blue plaque on the wall as you enter the building. This is a blue plaque for the work that was done at the MBA by Alan Hodgkin and Andrew Huxley for their work actually on a different cephalopod group. Squid, not cuttlefish, but they basically used the squid model to understand how nerve cells evolved. I'm going to come back to Huxley in a second. If we can have the next slide, please, David. So this slide here, I think, in my opinion, illustrates exactly what we do in the cell and molecular group at the MBA. So this is actually a really a representation of the work of Colin Brownlee and Glenn Wheeler and the work they do in their group. So on the left, you can see it's a satellite image of a phytoplankton bloom off the waters of Plymouth. And you can see this huge phytoplankton bloom dominating coastal water. Really important in terms of how the ecosystem functions. The organism that's causing this bloom is a coccolithophore. There's 2 coccolithophores in the centre image, and then the image on the right, the sort of the cartoon, if you like, on the right hand side, is a summary of the biological work that's been done at the MBA by Colin and Glenn and others to really understand the mechanisms of how these organisms work and function. And this really is the essence of what we do in cell and molecular. We take important marine organisms, they might be important biologically or evolutionary or from an ecological perspective, and we really understand their fundamental biology. This is an example here with coccolithophores, but we're doing this with bacteria and fungi and phytoplankton and a wide range of different marine organisms. Next slide, please, David. So we apply a broad range of techniques, actually, and even though this is a tech-centered presentation, I haven't got the time really to go through all the technologies that we use, but I think one of the categories of technologies that we use a lot is microscopy. And the microscopy work that we do at the MBA is innovative and contemporary. And I feel like innovative and contemporary are 2 words that so many people use now. It's sort of obligatory, but what do I mean by innovative? Well, we use microscopes not just to take pretty pictures of things, but we use microscopes to do experiments with marine organisms. So you can see here in the top left-hand picture, this is a microscope set up to do an electrophysiology experiment. David's already mentioned the meso lens, but this is an example of what I mean by contemporary, where the types of microscopes that we're using at the MBA are brand new and have not been used anywhere else before. So as David said, the meso lens, which is the microscope on the bottom left, this is a brand new type of microscope, and we're applying this now in the marine environment. If you can just move forward, David, some more of the image should appear. And David's already mentioned this, but I just want to reiterate, we have the new Huxley bioimaging suite, which is recently opened, and obviously for those of you that are paying attention, this is why we call it the Huxley suite, really to commemorate the work that Andrew Huxley did at the MBA. So next slide, please, David. So this is my last slide. I just want to mention what we do within Cell and Molecular Group in terms of education. So we're really keen to educate the next generation of cell and molecular marine biologists, and we do this through a range of different ways. We're host partners for PhD students, so we host for NERC and BBSRC doctoral training programs, so we have PhD students who come to the MBA to work. With the University of Plymouth, we co-run a Masters in Research in Marine Biology, and the students can choose to take a cell and molecular route in that and be part of the cell and molecular group. And then the MBA also hosts a range of advanced research workshops for people who are postgraduate and above to come and learn the different techniques. So that's it from me, David. Over to you.
Okay, David, if you'd like to pop your video on, or I can put you on there as well. Nope. Okay. You're on mute at the moment, David. So if you'd like to. Cool.
Sorry, I'm back. I'm back. Sorry, a slight issue there. I was on different screens. Okay. Is that okay, Mandy? Is everyone seeing
That's
that?
fine.
Brilliant.
We're now on the biologging of ocean predators screen.
Yeah. So thanks very much. This is the third of the case studies, and I just want to give you a brief sort of overview of some of the work going on at the MBA on biologging of ocean predators. It's myself leading a group of a large number of people working all over the world, actually, but some of us based here in Plymouth. And I guess the first question is, well, what is biologging? Well, telemetry and biologging science is really using these miniature animal-borne electronic devices for logging or relaying of data about the animal's movements, behavior, physiology and or environment. And there are lots of different types of tags that can be used to give us data on different aspects of the lives of these large ocean predators and sharks and seals and whales. And that enable us not just to understand the sort of fundamentals of their behavior and ecology, but also to give us ideas on where they are in relation to potential threats that they may encounter. Here you can see a range of different tags that we've used ourselves for tracking sharks. That's our sort of focal group, our focal taxa. But in fact, these tags can give information on all sorts of different things, not only the location. You can see here in the middle, there's a diving penguin sort of at the surface and then diving. These tags give location data, which can be relayed via satellite back to the laboratory. But also we can get, as well as movement data in sort of horizontally, we can get vertical data as well, their diving behavior, and that gives us sort of ideas about the depths at which they're foraging and what they might be feeding on. But as well as that, these tags can also give us amazing information about the conductivity of the water, the temperature, the depth, and even the physiology of the animal, heart rates, dissolved oxygen in the blood, and the blood chemistry of these animals. So not just behavior, not just ecology that's relevant to conservation, but also ocean observing. These tags are often they're so sensitive and high quality data that they can be used by oceanographers to inform about ocean temperature, for example, and oxygen and salinity. So a huge range of things that these tags can do. Ourselves, we've been tagging various species of sharks. Here you can see the blue shark. That's tagging going on in the Azores Islands in the central North Atlantic. And we attach tags to the sharks to track their movements and their behavior. And one of the reasons that we're doing this is to understand something about the space use of the animals, simply because sharks are declining. Many populations are declining. There's about 100 million sharks caught every year. And about half of those are these large pelagic sharks that range out from coastal waters right out into the open ocean. And pelagic longline vessels take millions of these sharks every year, and the populations are declining to the extent that we need to understand where the sharks are. You can't manage a stock unless you know where it is. And so one of the things we've been doing here, you can see in red the track of a blue shark that was tagged just about 20 miles off Plymouth by myself a few years ago. And we actually linked this to tracking of individual fishing vessels. And in fact, that fishing vessel track you can see in blue there actually caught this blue shark along its journey. And so that led us into a field of study where we started to look at not only the shark as a predator, but also humans as super predators taking these individual animals. And that led us to form this Global Shark Movement project, which is occurring at the moment. It's an ongoing project. There's about over 150 scientists involved across 26 countries. And really, this is tracking movements and changing habitats to quantify the threats that they face. And so we're able to bring all this data of these different species, great white sharks off South Africa and in the Pacific, with blue shark data from our own waters here in the UK and across the Atlantic, bring it all together to understand the areas where these sharks like to aggregate. Where do they like to hang out? And then we can relate that to the distribution of pelagic longline fishing efforts. So this is a map of where longline fishing vessel effort was located globally. And so the hotter the colour, the more fishing effort that's going on. And so you can overlay these two maps, if you like, and analyse. You can drill through the different layers to analyse where the hot spots of exploitation are happening. So here in this map, you can see the blue spots are actually where tracked sharks were caught. But the sort of hot areas, the hotter, gives us an idea of the overlap hotspots. So where the sharks are, or the vessels are overlapping with the sharks. And we found that about 24% overlap per month of sharks and longliners. And that can go up to sort of 76% for commercially valuable sharks. So as well as understanding where the main exploitation is happening for use in management and conservation, these tags also give us ideas of clues about the ecology and behaviour of the species. And here you can see some tracks. This is an oxygen map of the ocean. You can see tracks of blue sharks going into oxygen depauperate areas, these so-called oxygen minimum zone, these hypoxic regions. And we had to work with companies to design tags that would actually enable the tags to work at terrific depths. Here you can see a blue shark on the left and a mako shark on the right, diving almost every other day to depths exceeding 1,000 meters, often to nearly 1,500 meters depth, 1.5 kilometers down. And so the tags that we're using obviously have to be able to withstand those sorts of pressures, but also the cycling of that pressure change. And those sorts of tags give us clues as to other sorts of problems that sharks might be encountering. For example, these oxygen minimum zones are expanding, increasing hypoxia with climate change. And so it could be that the habitat of these sharks becomes more compressed in the future. And so they will be more accessible to these surface longline vessels. And my last couple of slides is actually just to give you a sort of heads up of some of the new tags that we've been developing with colleagues in Portugal. We've got a new project that's just starting next month, Ocean Deoxyfish, where we're looking at how sharks and tunas and other fish are interacting with these deoxygenated areas. And we've developed new tags that can actually record dissolved oxygen as the sharks and the tunas move along. It also records depth and compass heading as well, temperature and accelerometry. So we can get an idea of the body movements and tail flaps and tail beats of the animals. So we can start to look at how much it costs them to be in these oxygen depauperate areas. You can see here on the left, one of the tags that has been developed here has a wireless recharger and also pops off the shark, floats to the surface and then signals its position via GPS to shore-based researchers who then have to zoom out in a boat and locate it and pick it up, and then you can access the data on the right you see on this slide. So my last slide is just to our latest tag here is we don't just want to get a view of the zeros and ones, the sort of data that's coming through on temperature and dissolved oxygen. We also want to get a view of what the shark or the tuna is seeing at these depths. And this will then help us to understand the ecological importance of these climate-driven changes that are going on. So we have developed a video logging tag that also records depth, temperature, acceleration, speed, and will give us views on what the sharks are encountering and hopefully give us estimates of what they're feeding on and how often they're feeding. So incredible sort of developments are happening here where we're actually able to punch through that sort of mirrored plane at the surface of the ocean and really start to delve down through that window into what's going on in the interior of the twilight zone, the mesopelagic zone. So with that, that sort of concludes my biologging of ocean predators bit. But there's also a slide here which has been added by Rowena, actually. I don't know if Rowena, if you want to speak to this slide, it's the last one that we have, apart from our conclusion slide.
Thank you, David. Yes, this is just a brief slide just to let you know what the MBA can offer any interested people. We help the Marine Business Technology Centre offers helps to SMEs. As we've talking about, the continuous plankton recorder platform can offer you any kind of sensor or water collection facilities. We've got the sepia boat that can offer a variety of testing platforms. The seawater tanks, we're getting a new set of seawater aquaria, which will be state of the art. the microscopy suite, which was covered before, molecular lab and the culture collection, but also we offer services from consultancy on to people who are interested in a project but don't want some further information. We offer genetic testing and sequencing and the use of our long-term biological databases. And that's all I want to say. Thank you very much.
Super, thank you very much. I think we've got a little bit of time. I know we've slightly run over, but we were a minute or so late starting. So if anyone would like to raise some questions that have been listening to these very interesting presentations, do so now. None have come through during the presentation. I just need to give a couple of a few seconds because there is a slight time delay on them coming through. But if there are no further questions or no questions coming out of this, the presentations will be recorded so that people can access these at their leisure, check anything or go through anything that is of particular interest. No questions coming through, so it just remains with me to say thank you very much to our three superb presenters, working in an amazing facility at the MBA. So thank you all and have a very good day.
Thanks very much.
Okay, thank you. Bye-bye.